In order to see the impact of catechin and quercetin against cisplatin-induced in vitro cytotoxicity and altered antioxidant status in renal epithelial cells, we used NRK52E cell lines. These cell lines were basically procured from NCCS Pune, and once we received the cell lines, we incubated it further for a span of 24 hours. After 24 hours, these cell lines were subcultured into different flasks. Once the flasks reached a confluency of 97 to 99%, the flasks were further trypsinized using trypsin EDTA solution and were seeded in 96 well plate, which was further incubated at a temperature of 37 degrees Celsius and 5% carbon dioxide. After giving the toxicant, we further put the 96 well in the incubator. After an exposure hour of 24 hours, we put our dye, that is either MTT or ricesurine, and we further kept it for 24 hours in the incubator. After the exposure period, we put the dye of choice, either MTT or ricesurine, in those 96 well plates and further incubated these plates for 24 hours. After the 24 hours, we took these plates and we took the reading of the plate in ELISA reader. For trypan blue dye exclusion test, we seeded the cells into 6 well plates. These 6 well plates were further kept in CO2 incubator until they reached the confluency of 97 to 99%. Once they reached the confluency of 97 to 99%, we dosed them with cisplatin along with different doses of catechin and quercetin respectively. After a span of 24 hours, we took these cells and we counted them by using trypan blue dye. In order to determine the antioxidant parameter in the cells, we use the same 6 well plate as for trypan blue, and we diluted these cells in all the wells 5 times, and after dilution of 5, we used these cells as a sample for detection of different antioxidant parameters via spectrophotometry. The data, it was further analyzed by using GraphPad Prism and SPSS software.